stragglers come in.
I have a question.
What?
Are we gonna have PowerPoint presentations?
You mean for your, yes,
When though? On Wednesday?
the week before finals.
Oh, okay, good.
So that week before finals, we'll have plenty of time to have all the things done.
Yeah.
You've had speech before, right?
No.
Have you had speech? Speech?
Yeah.
Speech?
Yes.
Yes? Speech? You've had speech? Okay,
Yeah.
well, I expect them to do fantastic because
No, I haven't had speech.
they've had speech before.
Don't worry, he would have checked the file
So,
anyways.
Okay, but the see, thing is, if you've had the speech class, you know how to address an audience, because we're taught that in there. You were taught how to talk to your audience and give a presentation that is seamless and correlates directly with the information that's up on the board, right?
No, my speech teacher was the best.
Well, I took speech class as a, was it 6 weeks course? It was taught by a graduate assistant. Almost all of your classes and labs, most of your labs at the 4-year university are taught by graduate assistants. So it kind of makes sense that I would rather take a lot of those classes at the community college where they're actually taught by a professor, both the lab and lecture, rather than go to the lecture, you get taught by the professor, and then you go to the lab and get taught by the graduate assistant, who's, 2 years ahead of you in school. All right, so we got to the first one. Be able to describe a bacterium from the growth requirement given for it. So I give you several different aspects about their growth requirement. Their carbon requirement, their energy requirement, as well as their oxygen requirement, their state of being able to fight against toxic forms of oxygen, as well as their temperature requirement. I could have thrown in their pH requirement, such as acidophile or alkalophile or neutrophile. We know what those are, right?
Wait, can you, state of being able to what? You said carbon, energy, and oxygen requirement, and then you said state of being able to something.
Fight against a toxic form of oxygen.
This is my definition, and see if I have all this included. A chemoorganoheterotroph is
Yeah.
a bacteria that gets its energy source and carbon source from an organic molecule.
Yes.
Since it is facultative anaerobic, it prefers oxygen, but can grow without oxygen. Catalase positive bacteria will produce an enzyme for taking it from oxygen. Since it is a
Toxic form of
mesophile,
oxygen.
it prefers to live at 37 degrees for optimal growth.
Okay. So it produces catalase, which is an enzyme that will attack the superoxide or peroxide molecule, turning it,
What you're saying.
producing hydrogen as a consequence.
Wait, what?
Can you just say that it protects it from the toxic effects of oxygen?
You want to be very simple about it, yes.
Yeah, we want to be here. We've only got an hour to
But
find
it
all this
produces
stuff out.
catalase or something?
Catalase, it produces an enzyme catalase, right?
Yeah.
Catalase protects the bacteria from hydrogen peroxide. Remember when we put hydrogen peroxide on the
Yeah.
gram-positive staff and it bubbled up? And it is a mesophile,
What's it?
meaning what?
I prefer to live at 37 degrees Celsius.
Optimally at 37 degrees Celsius, but it can live at 20 degrees Celsius and 45 degrees Celsius, but it grows best at 37 degrees Celsius, which makes it medically important for us, right? Because our body temperature is at 37 degrees
It can
Celsius.
live at, what?
It can live anywhere between 20 degrees Celsius and 45 degrees Celsius, but it prefers to be at 37 degrees Celsius because that's where it grows optimally, is best.
Got it, I got that.
And 37 degrees is our body temperature, which makes it medically important.
I think this is good. I
Hmm?
think that's the only question.
What'd you get?
Just like s'mores stuff.
Oh,
And then coffee.
I can't do the fruity stuff, oh, not fruity, but the extra sweet stuff anymore.
That's too
Okay, everybody good on the first one? Okay, second one, describe in detail the formation of a biofilm from planktonic state to maturity. Anybody do that?
Biofilms are formed at planktonic state where microorganisms are free swimming. They use their membrane and slime layer to stick to surfaces.
Okay,
They use quorum sensing DNA and proteins to communicate and bring in more organisms.
yes.
Channels start.
Organisms can be different species.
So can we just say organisms or different species?
Yes,
Bring in-
you can, it brings in their own organisms as well as it attracts other species as well.
Okay. And then I have channels start forming to bring in water and nutrients and let waste out. As the biofilm matures, organisms will break off and start the cycle over.
Okay, that's good.
Does that cover our
That covers,
requirements?
yep. Okay, number 3, describe in detail the typical microbial growth curve. Describe the events that occur in each phase. So, what are the 4 phases of the growth curve?
Your lag, your log, your stationary, and your depth.
Okay, so this growth curve is an example of a closed system, right? Meaning there's no entrance of nutrients and there's no removal of waste. So they're gonna go through this typical growth curve. In the lag phase, what is occurring at this point?
Your culture is adapting to its environment.
So it's adapting to its environment. Is it metabolically active?
It is metabolically active, but there is no
Population growth.
Right.
There is growth of the bacteria in size, but they are preparing themselves for division.
So they are metabolically active but are not multiplying.
Yeah,
Just growing.
they are growing in size to prepare themselves for division, but they are not growing in population at this point. Okay? Now, the log phase, what's going on in the log phase?
Your exponential growth.
Okay, so you have exponential growth. What is the nutrient status at this time?
What do you mean?
The nutrients that the bacteria use to live, what's the status of that nutrient?
Has a lot of nutrients.
It's in high abundance, right? What about the metabolic waste at this point? Question?
Very low.
Yeah, it's minimum. So you have exponential growth of the bacteria because there is plentiful nutrients and minimal waste to be toxic to them. Okay, now the next phase is stationary. What's going on in stationary?
The nutrients are declining and the metabolic waste is increasing.
Okay, so you have a declining of nutrients and an increase in production of waste. So that what that is causing is an equal amount of, growth as far as population and an equal amount of death. So some cells are dying off, but they're dying off at the same rate that they are being produced.
So can you just say, like, you have an equal proportional to growth to death?
You can say that. Okay, the next phase is the death phase, or the decline phase. So what's going on there?
More death and growth.
So you have an unequal proportion of death to growth, meaning that you have more dying off than you have multiplying. And so now we look at the nutrient content and the waste content and how that's affecting, right? At this point, the nutrient content is very minimum, not a lot left, and the amount of metabolic waste is at almost a maximum, so it's killing off a whole lot more as well. Not only are they starving, but they're also, some of them are also dying because of the toxic effect of the waste. And at this point, you're going to have, the ones that are still alive are going to be pretty hardy, meaning that they have been able to genetically change over to use a different carbohydrate that is in there. If glucose is used up, they can switch over to do lactose fermentation or whatever they need to do to survive. Okay. Describe in detail the process of performing a serial dilution and viable plate count estimation for microbial population size. Okay, so you're doing a serial dilution. Number one, you gotta figure out, why am I doing a serial dilution to get a viable plate count?
To isolate, to get a better isolation of the liquid colony.
Yeah, your goal is to get a better isolation of colonies from the sample that you have. So you start off with a sample I gave you for your unknown. It's highly concentrated. So if I wanted to get a viable plate count of them, I could do a serial dilution of that sample. If that sample is 10 milliliters, and I want to start diluting it, I'm going to have media in several other tubes. And that media amount or volume is going to be 9 mils. So to properly do a serial dilution, I'm going to take 1 mil of my starter solution that contains the bacteria and put it into 1 tube of 9 mil. That now makes the volume at 10 mil, right?
Mm-hmm.
So the ratio of dilution at that point is 1 to 10, right? What happens if I plate that stuff? I still have too many to count, too many colonies to count. So I'll do a successive dilution. I will take 1 mil of that diluted solution and put it into 9 mils of broth. Okay, now I bring the volume up to 10 mils. Now my ratio for dilution is what? 1 to 100. And if I plate that, what is going to happen? Still too numerous to count. Okay. So now I'll do another dilution. Take 1 mil of that solution, put it into the next tube. What's the ratio there? 1 to 1,000. If I plate that, what do I get? Around 65 to 100 colonies. Still not really isolated. If I really want to isolate it, I'll take another dilution. I'll take 1 mil of that solution, put it into another tube. What's the rate of that one? 1 to 10,000. And how many colonies do I expect there? Around 6 or so. Okay, so I've been able to really isolate that bacteria at that point. And so I would be able to take one of those colonies and do whatever test I need to do, whether it be a gram stain or start a subculture of that, because I'm going from a pure culture to subculture, right? So you can do a lot of things with it at that point. That's why we would do that type of serial dilution. Yes, ma'am?
Do you take the milliliter out of, like,